So let us learn about the practicals of genetics for students' convenience. I'm just giving the brief introduction about the different experiments. So first experiment is to study the Mendelian inheritance using suitable examples. Verify the result using Punnett square. So all of you know about the Punnett square and Mendel's law. So how Mendel's used to give different kinds of the laws here. So first of all, the first law, or how he used to selected the pea plant for explaining the different concepts. So Mendelian inheritance, there are the terms, two terms, monohybrid cross and dihybrid cross. So first of all, first you must know about the monohybrid cross. So when only one character is taken, then it is the monohybrid cross. Like example is given here, pure tall plant designated by capital T and capital T crossed with the dwarf designated by small t and small t. So you are getting the hybrid. One is capital T and another is small t. So you are getting the hybrid one. So when these hybrid T, capital T and small t and capital T and small t, they are self-crossed, then gametes are formed, same kind of gametes, capital T, small t, capital T and small t. And when they are self-crossed, you are getting the ratio in Punnett square here. This is the Punnett square. You put the gametes here, capital T, small t, capital T, small t. And after crossing, you get the results here, like capital T, capital T, capital T, small t, capital T, small t, and small t. So this one is showing the tall. These two are also the tall. The hybrid are the tall. And only the dwarf is the uh, small t. So ratio comes when you see the genotypic ratio. This is the 3:1. 3 tall, 1 is dwarf. But phenotypic ratio, when you see the genotypic ratio, when you see the genes, then it comes to the 1:2:1, right? Phenotypic ratio and genotypic ratio, you should know about the phenotypic ratio and genotypic ratio. So this is the first experiment. So genotypic ratio, you have seen here, one tall, two intermediate and one dwarf. So these are phenotypic and genotypic ratio. So you can just go through this experiment by taking the one example. This is the monohybrid cross. Then in second, you have to explain the dihybrid cross. So in dihybrid cross, two characters are taken, like round color, round seed and yellow color, wrinkled seed and green color. So capital R, capital Y, small y. The gametes are formed, capital R, capital Y, small y. And in F1, hybrids are formed, capital R, capital Y, small y. When these are self-crossed, so in this way, when these are self-crossed, you are getting here the gametes, and in Punnett square you can put like this, you are getting the results. So capital R Y, R capital R small y, small r capital Y, small r small y, like this on the left side also, and you are getting the results here. So when you are getting the results here, you can see that 1 2 3 4 5 6 7 8 9 are of same, 4 are same, and 3 are 9:3:1. So means 9 3:3:1. So ratio is 9 is round yellow, and 3 is wrinkled yellow, and 3 is round green, and 1 is wrinkled yellow, which is small r small y. So in this way you can explain the dihybrid cross and the monohybrid cross. So this is about the two experiments, monohybrid cross and dihybrid cross. So likewise you can explain the epistasis here, when one gene character hide the effect of another at other locus. So you have to give the examples here, and you are showing the Punnett square then, and in Punnett square, after you are getting the result, white 12, gray 3 and black 1. Black again uh, 3. 12:3:1. Black is 1. So in this way you are getting the results in epistasis. Results changes from the Mendelian ratio. So this is the non-Mendelian ratio. So these are the three experiments: monohybrid cross, dihybrid cross, and about